Started on coding, are there any questions about the ANOVA report? Presumably you've all finished your ANOVA problem set. Or for that matter, did you have any questions about the ANOVA problem set? I didn't get any emails from anybody except one person on the ANOVA problem set. Was that fairly straightforward? How did you guys handle the last question? Was that easy enough to figure out? You were able to figure out, oh, he's just talking about variances. Variances are what goes into the ANOVA, and you were able to figure your way through that. Okay. Well, good. That last question was a question that was designed to make you think about what exactly an analysis of variance is doing to kind of reinforce this idea of how the analysis of variance actually functions, because that's important to know. So has anybody started on the report? Okay, so as you start working through that, if you have questions about the code that you need to generate in order to do that, etc., once again, email me, schedule an appointment with me, whatever. Remember that the template for how the report should be structured is on the Moodle site, and we're going to finish talking about R Markdown today, and then after we're done with R Markdown, we're going to start talking about a two-way analysis of variance. So even though you're still working on a project having to do with a one-way ANOVA, we're going to move on and start talking about two-way ANOVAs. So if you have your P's analysis script up, if you hadn't cleaned that up last week in preparation for going into R Markdown, hopefully you went through and cleaned up and organized and sectioned and annotated that script, because it'll just be useful to have all that done before you start transforming that script into an R Markdown document. So in my, oh, that's not the right one. Where is my piece? There's my piece Markdown file. So I've got my P's Markdown file, and I've got it shown all the way through the end of the data transformation phase. So if we go back to my P's analysis file, I've loaded in the documents, I've read in the data file, and I've cleaned it up. And what I mean by cleaning it up is when I expand this out, I read the data in, I get a sense for what is in my data. I make sure that R is treating my data as a factor. The columns that I want to be treated as a factor, those are treated as a factor. I then have told it that I want it to maintain the order that those treatments are listed in, so that it does that. And then I check to see if it actually is doing that. Go back to my Markdown file. I've also tested the assumptions. So the first thing that you want to do when you want to go and test the assumptions, going back to my analysis file, is you have to specify the model, which only takes a line of code in reality. This is just saying my model is an analysis of variance. It has treatment as the independent variable, and it has growth as the dependent variable. And then my next section of coding is all about testing assumptions. And at this point, I test the assumptions, and I find that we don't have homogeneity of variances, and so then I apply a data transformation to my data. And in the Markdown file, that's where we ended up with. I forget who I was working with last week. Who was I working with last week? Oh, I was working with Blaine last week, and we were getting him kind of caught up. He was having some difficulties with his script during class, and we didn't have time to figure out everything that was wrong with it, so he came by my office. And one of the things that you'll notice when I'm scripting is that when I do an R Markdown document, you will see that I use big chunks. So remember that in our Markdown, we refer to these things as code chunks. How can we pull up a code chunk? What are the two methods for beginning a code chunk? Pardon? Command Option I, that's new for me. So one way is Command Option I. I'll have to add that to my list of shortcuts. What's another way? What's every code chunk begin with? That you can just type in yourself also. Here's the beginning of my code chunk. What does it start with? Yeah, they're not apostrophes, they're backticks, but it's the non-shifted tilde essentially. So 3 backticks will get it done. What's the other way that you can do it? You can also just go up to code and insert chunk. So I haven't tried this thing that Chance just suggested. Command Option I. So Command Option I is just the same thing in keystroke form as insert chunk. The thing that I like about inserting a chunk is that it gives you the 3 backticks. It gives you the little R in brackets, and it gives you the ending 3 backticks for your code chunk. If you just type in the 3 ticks, you then have to remember to put R in brackets, and at the end of your code chunk, you have to remember to put the 3 backticks. So my advice always is to do things in ways that R is going to do as much of the work for you as possible, because that just makes things easy, because you don't make mistakes because you forgot to put the final 3 backticks. Now, for this particular code chunk, we could have divided this thing up into lots of little sections. So we could have done one section where we test the where we generate the residuals for the model. Then we could have had another code chunk, Command Option I, where we then do the Shapiro test on this. And this will work. And then we could, after that, make a little code chunk where we then build a histogram for that. And so in between these things, we might want to put in comments. So for example, we might put in a comment here, and that just tells us what we're going to do in the next step. So we've already got generate residuals. Remember that the number of pound signs that you put before text determines ultimately how big that text is going to appear in terms of font size. And we could do that through this whole thing. And so it would be a bunch of little comments separated by little chunks of code that all kind of do this thing. In my version of doing it, I've put this all in one big code chunk, and within this code chunk, there are comments. Now, one of the things that you'll notice is that here's a comment down here at line 118, and that comment is about Levine's test and demonstrating that Levine's test really is just a particular type of an ANOVA. And so if we go down to my original script, you will see that that is commented out with a pound sign in front of each of those things. Turns out Command-Shift-C, as you go Command-Shift-C, it will get rid of all of those things. So you can comment out multiple lines of text. Command-Shift-C puts those things back in there. Command-Shift-C gets rid of them. So taking these multi-lined comments like this in your Markdown file, it is easy enough to just undo them all. So take that as a comment versus not take that as a comment. Now, one of the things that you'll see when it does that in our Markdown is it just puts a pound sign at the very beginning of that comment, because in our Markdown it's going to auto wrap that. If we just took this thing here and copied and pasted it into our Markdown file as it is, and I'll just do it right next to it, you're going to see that it treats that differently because there is a wrap function in our Markdown, and as a result, it's going to wrap that text around, which means it's going to be broader in your R Markdown file than it is in your R script. And as a result, these little hashtags, these little pound signs are going to appear in the middle of lines in your R Markdown file. So I'm just going to knit this. Whoa, what happened? I have a duplicate chunk somewhere. Which duplicate chunk is it? It is this one. Try this again. So here's my R Markdown document that comes out of this. And when I get down to this section where we're doing things, this is the first section where I test the assumptions of the model. So I generate the residuals, then it gives me the output. I conduct a Shapiro-Wilk test. I made some typing mistakes, not surprising there. I generate the histogram. And then I run a Shapiro-Wilk test. Then I generate the residuals again. So at this point, this is not in any section itself because I had to get rid of that because I have redundancy in my headings. But this is now doing the same thing. And when I get down to the point where I test for Levine's test, I have two ways of doing it. I have this way of doing it, and I have this way of doing it. And in this Markdown view here, it shows it all still parsed out the way it is in your script. But if you go to your Files tab, one of the things that you will find is there should be a P's Markdown document text. This is a Word file. Go and open up your Word file, and it will show you what the document will actually look like in something like Word. And as you can see, I have one big chunk of code here that is about examining your data. And within that code chunk, the comments appear just like they appear in the R file, the R script. And then we have the testing assumptions of the model, where I've generated the residuals from the model, and then I have testing the residuals from the model, and then I have viewing the data in a histogram, as opposed to when I do it in one big code chunk. Wait a minute, what happened? I know what happened. I didn't knit this new version yet. So go back and knit to Word. under the knitter thing, it's going to then overwrite that document. Yeah, here we go. It helps if I pull up the right thing. So when I get down to this section where I'm testing the assumptions of the model, this is the portion of the code where I've busted it up into little tiny code chunks with comments in between each one. And so here's the first comment, here's the second comment, and then here's a section where I have broken it up into one big code chunk, and these things are actually just commented within R. And when we get down to the section where oh, it didn't do it. Okay, so I guess because these are really long, it doesn't do it this way. But in some cases, when you knit this to a document, this will appear as a long line, and then this pound sign will be embedded in that line somewhere, and it just wraps around itself. It hasn't done that in this case, probably because I made my comments long enough to fill the width of the page. But I was doing something the other day when it did that, and it was irritating. So I would advise you to just put a single hashtag in front of these long comments, and then they appear as little paragraphs. Now, the reason that I like doing bigger code chunks is that if I was doing this in little code chunks, if I was doing this code chunk and then this code chunk, each of these comments would appear as a standalone title, a standalone section heading. In the same way that up here, these things are standing alone as standalone section headings. Section heading, section heading. This is technically a section heading. This is technically a section heading, but you can see that I didn't leave a space in there, and as a result, it read it differently than if I had left a space in there. It appears in a different color, etc., etc. So one of the advantages of doing larger code chunks is there's less post-markdown editing that you have to do. So if I was going to fix this, right here, I would have to go back to my R Markdown file. I would have to put in a space here. I would put in the O there, and then I would reknit this to Word. And when you reknit it to Word, it's going to give you the Word file back again. But the Word file has now been altered. And this thing now appears as a heading. And that's fine and everything else. But you'll notice that this subheading is a different font size than this subheading. And the reason that it's a different font size is because in our Markdown file, this has 3 pound signs in front of it, and this one has 4 pound signs in front of it. So once again, in order to get a document that has a uniformity to it, one of the things that you would like to have is to have all your headings kind of in the same font size. Headings and subheadings, kind of like you would have in an outline. If you're busting your R Markdown file up into lots of little chunks with comments in between them that appear as headings, you have to then go back and be fairly rigid about how you're formatting things, as opposed to bigger code chunks where all of the annotations that you have from your original document are just appearing as annotations. It ends up looking a lot cleaner in the final R Markdown section. So go back to the Word file. Here's the beginning of the long block, the big code chunk. It starts with generating the residuals, and then it gives you what the residuals are. So it's going to give you the code, and then it's going to give you the output. Then you have the code, then you have the output. This ends up looking much more like an R script would appear. Here's the code, here's the output. One of the advantages of doing little chunks, little code chunks, is that when you run, well, it's not really doing it that way, is it? Let's go back and knit to HTML. One of the nice things about knitting to HTML is that in HTML, it gives you your code in gray blocks, and it gives you your output in these open blocks. And so if you ever go to like Stack Exchange, let's say we want help doing a two-way ANOVA. When you look at anything in Stack Exchange, the people who are interacting in Stack Exchange often are doing things in well, let's not do this. Let's go to there's just as a resource for you, there's this amazing guy named William King at Coastal Carolina who years and years and years ago started a blog basically kind of documenting his life doing learning programming in R, and it's very useful. It's a very useful website if you're trying to learn how to do things. So let's say that you want to learn how to do a simple t-test. Whoa, not found. Well, his stuff isn't done that way either. Nah. Ah, here we go. This is going to be one of those things. So this is an R Markdown file basically that has been put on the web. And one of the things that you can see with this R Markdown file is that it has these little code chunks that are separated by formatted text. This is what an R Markdown file looks like when you see it on the web. And so one of the nice things about knitting to HTML to view it while you're working on it is that it clearly shows you the difference between what is code and what is output from code. That is less clear in the Word document. You have to actually pay attention to in the Word document where you have pound signs. So this is getWD, that's the command. This is the output from that command. Once again, head.dat is the command. This then is the output from that command, and you know that it's output because it has the two pound signs in front of it. So it's easy enough to see once you're kind of attuned to what R is doing. And you'll see that that's very similar to what R actually looks like if you're going to go and read in a data file and then get its structure. You don't see those hashtags in front of this, but in front of each of these lines in your R Markdown file, there is two pound signs indicating to you that this is model output. This is, you've asked R to do something, and then it is given you the result of whatever you just asked it to do. So R Markdown is not all that complicated. The nice thing about it is if you're looking at it digitally, is it color codes things. So in text or in script, annotations appear in one color, headings and subheadings appear in another color, et cetera, et cetera. Things that you specify, like levels and data, appear in a different color. And the other nice thing about R Markdown is that when it comes to generating things like your figures, they're already just naturally embedded into the document. So I asked R for a histogram, and then this is the histogram that it gave me. So R Markdown is not very complicated once you kind of get the hang of it. My advice is to build your script in regular R, make sure that it's functioning, make sure that it's doing everything that you want it to do in just regular R, and then transfer that to a Markdown file. Now what this means is that in order for you to do this report, you can't be throwing this together the night before it's due probably, because you need to have been working on the script that actually does the job of analysis far enough out in advance that you have time to run the analysis, write the report, and prepare the Markdown file. So this is definitely one of those things that if you haven't started on it yet, you need to start on it because it is going to take some time. The analysis itself is not particularly complicated, but getting the write-up done in a way that makes sense to you and will make sense to me will take you some time. And then just the physicality of transferring what is an R script into an R Markdown document can be irritatingly niggling sometimes. Your goal in preparing all of this is assume that I am a person who doesn't know all that much about statistics. I know some. Maybe I've had an undergraduate course in statistics 20 years ago when I was an undergraduate, and I've asked you to analyze some data from me. You need to, in your report and in your R script that you give me in Markdown form, you need to be walking me through the analysis in the most logical way possible, and you need to be annotating that script in a way that when I receive that script, it's walking me through the analysis and telling me what you're doing at any given time and why you're doing it, in a sense. And so in all of my R scripts that I prepare for this class, I'm usually much better at doing this for things that I prepare for class than for things that I prepare for in my own work. But you'll see that my R scripts are amazingly heavily annotated. It's a habit that I get into when I'm doing things for you guys in class because I want you guys to understand what it is that I'm doing and why I'm doing it. I tend to not do that as much for R scripts that I write for myself, but in reality, I probably should, because there have been occasions when I've done something in analysis and I come back to it 6 months later because it's time to write the paper, and I get to a line of code and I'm like, why did I do that there? Whereas if I had just given myself an annotation at the moment saying why I did that thing that I just did, I wouldn't have to ask myself that question 6 months later. And so the act of annotating your code becomes very important, not only for you so that you know why you did the things that you did when you come back to something after it sat and rested for a little while, but then also for anybody who's going to use your code to do a similar kind of analysis, then they know why you did the thing that you did, even if you yourself can't remember it. All right. Other things about the report, our Markdown, etc., etc. Ultimately, you're going to save the Word version of our Markdown, and you can either submit that to me as a Word document or a PDF. I prefer PDFs because that's how I'm going to look at it on my iPad, but if it's a Word file, that just means I have to go through the extra step of saving it as a PDF and then looking at it on my iPad. Questions about all that? Yeah. Next Monday, I think, is the due date that's on the Moodle site. Yeah. So you already rely on the script that you already have for doing an ANOVA, because it's a script that has enough problems with the data. For example, it had to do a data transformation. In this case, with the gecko body temperature data, all of those data are very normally distributed, so you shouldn't have to be doing any data transformation with that. So that's the nice thing about it. But if you do, once again, there's a section in the script that you've prepared for the P's analysis that deals with data transformation. But take this order. Whenever I'm thinking through a project, there are a set of steps that I need to go through. Basically, this is the steps of any analysis. Get the data in, check it to make sure that it's the data that you thought it was. Check the assumption of the data. If you violate any of the assumptions, then do any transformations and then recheck the data. I should put recheck assumptions once you've done this. No point in transforming your data if you then don't go back and check the assumptions after you've transformed the data, because maybe the data transformation that you tried doesn't actually fix the thing that was wrong with your data. Once your data are in good shape, you run the analysis, you conduct any post hoc tests that are necessary. Once again, if you ran an ANOVA and there's no differences, then there's no point in running any post hoc tests because you're going looking for differences that don't exist. But run post hoc tests if necessary, and then produce whatever meaningful graphics you want to produce to illustrate what the results of your analysis were. One of the things that I complained to my girlfriend about is she's very good at graphics in R because in genomics they use a lot of graphics. And I complained to her because it takes me 5 minutes to run an analysis and 3 hours to produce a set of graphics that I'm happy with. And so once again, making these graphics so that they're intuitively obvious to the person viewing the graphics as to what the graphics mean, making them aesthetically pleasing, et cetera, that can take some time. That can sometimes take an irritating amount of time. So I'm usually pretty quick through these first stages of analysis because that's kind of how I was trained. And the first statistical packages that I learned on, they generated graphics for you automatically, whereas in R you have to specify every little niggling thing about a graphic. And so this always takes me more time than all of this up above does typically. So don't forget to leave yourself enough time to generate meaningful graphics. But this is any analysis on the planet. This is what you do. Get the data, check the assumptions for whatever test it is that you're running, run the test, do anything post hoc that you need to do to tease apart what that result is about, and then creating graphics that have to do with that. And that's every analysis you will ever do the rest of your life. It follows the same format. I would organize your R script in a way that kind of follows that same pattern. Now, there's potentially lots of steps within each of these things. Obviously, between running analysis and producing meaningful graphics, you need to summarize your data using summary SE. So there's stuff that goes into each of these steps. This is the pattern in broad scales. Questions? Yeah, Sam. Yeah, so for homeworks, I generally have them do at the beginning of class. But for assignments like reports and stuff like that, I typically do them at the end of the day. I don't remember what the time was that I put on the report, but I'll check that later on today after I'm out of class. Yeah, generally end of the day, so if you have any problems in getting it together. Homeworks are worth less than the reports are, so I want you to have every opportunity to get the report right. So yeah. So if it's not got a timestamp on it, or if the timestamp is wrong, I'll fix that. In my biology classes, we separate preparatory homework from practice homework. Preparatory homework is always due at the beginning of class because it's homework that we ask you to do in preparation for class. Practice homework is practicing things that we've already gone over in class. So it's important that you do one before class starts, and it doesn't really matter when you get the other one done. And so we usually have a before class due date for practice or for preparatory homework and an end of the day deadline for practice homework. So it's kind of similar in this way. All right, other questions? All right. On the middle site, there is PowerPoint. And so what we're going to start with now is we're going to start learning a new technique. This new technique is an analysis of variance, but in this case it's a two-way analysis of variance. And it's a two-way analysis of variance because you now have two independent variables. So So in a one-way analysis of variance, remember that we had this is the way R talks about this. We basically have an independent variable that has some effect on a dependent variable. And in this case, there's only one independent variable that is influencing the dependent variable. And so the independent variable is independent because it can vary freely, and the dependent variable is dependent because its value is dependent upon the independent variable. And in a one-way ANOVA, there's just one of these. In a two-way ANOVA, there's two of these. The cool thing about the difference between a one-way ANOVA and a two-way ANOVA is that once you've ramped up to two independent variables, you can have a three-way ANOVA, a four-way ANOVA, a five-way ANOVA, a six-way ANOVA, a seven-way ANOVA, a 15-way ANOVA. It would be ridiculous to have a 15-way ANOVA, but you could, and the math is all still the same. So these things are easily expandable once you make the jump to a multi-factor ANOVA. So this is information that is found in section 9.2 of your textbook. There's a section 9.1 which talks about nested analysis of variance, which we will not be doing in this class. So we'll skip to 9.2. 9.2 is a long section of the textbook with lots of subsections. That whole thing we will be working our way through. So if you haven't started reading that yet, you need to also be reading that as well. So just a biological example of what this looks like. There's a place over in Kansas near Manhattan. It's a field station that is run by the Kansas State University. It's called Kanza Prairie. They're doing this big experiment, 8,600 acres. And in this experiment, they are altering two things. They have one section of the area where they have bison grazing, so they have native grazers. That's what the N is for. And then the other thing that they have is they have frequencies of burning return frequency. So prairies traditionally in the Midwest are maintained by fire. Well, you can have fires that occur frequently, or you can have fires that occur infrequently. So in this area where these bison are, they have plots, watersheds where they burn it every year. They have plots where they burn it every other year, every 4 years, and every 20 years. And so they have these two independent variables, the presence or absence of grazers and these different frequencies of fire. And so in this case, we have native grazers, which are present or not present. And so that's one factor with two levels. And then with fire frequency, we have 1 year, 2 year, 4 year, and 20 year burn frequency. So this is another treatment with 4 levels. So when we think about degrees of freedom for these two treatments, this would have 1 degree of freedom, and this would have 3 degrees of freedom because it's always 1 fewer than the number of levels that your treatment possesses. So we can already start to get a feel for what this experiment looks like. A paper that we read in one of my biology classes, it only deals with 1 and 4 year burn frequency, so this would now be a treatment with 2 levels, so the degrees of freedom would be 1. Doesn't matter, it just that in this particular paper that they were working with, they only looked at 2 of the burn frequency treatments. So what an analysis of variance in this case asks is, what is the effect of grazer presence or absence on whatever the response variable is, and what is the effect of fire frequency on whatever the response variable is? But because they have all combinations of this, they have some plots that have bison and 1 year burns, they have bison and 4 year burns, and they have no bison 1 year, and they have no bison in a 4 year burn. They can not only look at how each of these things affect, for example, species diversity, how many species live on a particular plot of land, they can look at the effect of bison versus no bison. They can look at 1 year versus 4 year, but they can also look at whether or not these things interact with one another. Does putting bison on a plot that is grazed every 4 years, how different is that from no bison 1 year? And is it simply the additive effects of bison versus not bison and 1 year versus 4 year, or is there some multiplicative effect? Having bison every 4 years, does that make a bigger difference than just the additive effect of having bison versus no bison and having 4 year burns versus 1 year burns? Those two things added together might give you some effect on species diversity, but those two things together might actually give you a bigger effect than the additive simple effects of the two variables by themselves. In which case, that then is the interaction term. The two treatments interact in a way that it gives you something that isn't simply the additive effects of each of those treatments by themselves. So one way of thinking about this is, in this particular system, they have these watersheds. So they did these in the native watersheds, 1 here, and 1 here, and 4 here, and 4 here. So they just did it this way. And rather than setting up another watershed someplace else where there are no bison, They just created pens inside of these bison areas where the bison were fenced out. So within this watershed where there's normally bison, there are little sections of the watershed where they've been fenced out. And then some of these are burned every 1 year and every 4 years. And so this is one way of thinking about this. Another way of thinking about it is you have your two treatments, grazing and burn frequency, ungrazed versus grazed, where bison are present. And then we have burn frequency, where it's frequent versus less frequent. And these are kind of graphic visuals of what their experiment looks like. And so one way of thinking about this is 1 year frequency different than 4 year frequency? That gives you the main effect for burning frequency. You can ask, are grazed areas different from ungrazed areas? That gives you the main effect of grazing. But it's the pattern from here to here that gives you the interaction. Within this, adding bison does a certain thing. Within this, adding fire or removing fire does a certain thing. But what happens when you both remove fire and add bison? Is that different than just the additive effects of adding bison and the additive effects of reducing burn frequency? And it's that interaction that is actually the important thing that people are interested in. As much as the individual effects, often people are really interested in the interactions. So, these are the equations. Now, what is my advice about equations? I've given you this advice before, so many times. It shouldn't bear repeating, but evidently it does. Pardon? So you can break it down by terms, but I have even better, more consistent advice. Am I ever going to ask you what the equations are for working in one-way ANOVA? No, why not? Can you remember them? Do you guys know them? But you've already done a homework assignment with ANOVA, and you're about to embark upon analyzing a set of data with ANOVA. What matters? That you can jot the equation down on the board? What matters? What matters is, can you do it? What's wrong with you people? Wake up. Wake up. Can you do it? Is knowing these equations going to help you do this? Probably not. I don't kid myself that it will. Don't kid yourself that you think that it will either. But these things we'll come back to and say, okay, now that we know how to do a two-way analysis of variance, how do these equations relate to that? So that the equations make sense to you. But my advice is, don't ever think that just knowing an equation is going to tell you how to do an analysis. These kinds of tables are useful because it helps you know what your degrees of freedom are and things like this. But in terms of this stuff, that's very difficult to say, okay, well, this is the row. So to get the sums of squares, it's the row mean minus the grand mean squared multiplied times the sample size of the rows times the sample size of the columns. We could do that, but I'm sorry, not the sample size. The sample size of each of the rows times the number of rows that you have. Sample size of each of the columns times the number of columns that you have, et cetera, et cetera. So that works if you know what you're doing. It doesn't necessarily work if you don't know what you're doing. So let's talk about what it actually does. So here is an example. We have two treatments. It's a simple example, which means that it's easily worked by hand, which is what we like doing at the beginning. Fat type that is fed to rats. We have fresh fat and we have rancid fat. So this is a treatment with 2 levels, which means that we have 1 degree of freedom. We have the sex of the rats that we're feeding to this, male and female. Once again, this is a treatment with 2 levels, which means that we have 1 degree of freedom. And what we did was, well, what somebody did was they took 6 male rats and 6 female rats. They fed half of those rats fresh fat, and they fed half of those fats rancid fat, and they looked at how fast those rats grew. And these are the data in terms of weight gain, probably in grams over some period of time. So one way of thinking of this is fresh fat, rancid fat, males and females. We have 3 observations here, and we can take the mean of males fed fresh, and we can take the mean of males fed rancid. We can have the mean of females fed fresh, and we can have the mean of females fed rancid. So why does it just fritz out like this periodically? I do not know. So remember that an analysis of variance is an analysis of variance. It is not an analysis of means. So I do not know what's going on there. So we have different values here. 3 values here, 3 values here, 3 values here, and we have 3 values here. Okay? So we have 3, 6, 9, 12 values total. So we could also add up all of these values together, regardless of whether it's fresh or rancid, regardless of whether it's male or female, and we could calculate the grand mean. If we add all of these up, the grand sum, if you add all of these things up, the grand sum is 71.73, and if you divide that by 12, the grand mean is 597.75. So in a one-way ANOVA, if we wanted the total sums of squares, how would we get that? If we were calculating sum squares total in a single factor analysis variance, how would we do that? How can we use that knowledge to tell us how we would do that in this situation? Sum squares total. So one way to do it would be to do sum squares among plus sum squares within. But of course, this is now more complicated. So what's a more direct way of doing it? That's absolutely correct. It's just not the answer I'm looking for at this particular moment. This is the grand mean. Sum squares total is asking, how does the grand mean differ from what other figures in this table? Is it asking, is it different from the column means? Is it asking, is it different from the row means? What is it asking, is it different from? Or how does something vary from the grand mean? What is that something? It's not asking, how does this mean vary from the column means? It's not asking, how does this mean vary from the row means? That would be sum squares of rows and sum squares of columns. Sum squares of total asks what? How does this grand mean differ from this data point squared? How does this mean differ from this data point squared? How does this mean differ from that data point squared? How does this mean differ from that data point squared? this from that, 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 all added up. That's the total amount of variation. It's how every data point varies from the grand mean for the entire data set. That's why it's sum squares total. So one way of writing this is basically, how does each value differ from the grand mean squared, summed across all rows and summed across all columns? That's what the sum squares total is. But that's just a fancy way of writing. How does this data point differ from the grand mean squared? How does this data point differ from the grand mean? Square that. This different from that squared. This different from that squared. This different from that squared. This different from that. All those squared deviations from the grand mean added up, that's the total amount of variation that is in your data set. So that total amount of variation is some value. And when we calculate that value, it ends up being 75,570.25. So if you subtracted the grand mean from this and squared that, subtract the grand mean from this and squared that, subtract the grand mean from this and squared that, from this and squared it, grand mean minus that, grand mean minus that, grand mean minus that, all those things squared, added up across the whole thing, this is the answer you would get. Sum squared total. Now, all that variation is due in a one-way ANOVA way. Sum squares total is due to some residual variation, sum squares residual, sum squares error, just noise in the system, variation around the means. This is sometimes also referred to as sums of squares within sample variation. It all means the same. Remember that sum squares within, sum squares residual, sum squares error. These are all the same thing, just different labels for the same thing. That variation is ultimately asking to what degree, now I do kind of need you to see this thing. Sums of squares within is looking at within males fed fresh fat. How does this value differ from that mean? How does this value differ from that mean? How does this value differ from that mean? All of those things squared and added up for this, for that, and for that. That's the within sample variation. How much variation is there within each combination of the two treatments? That's what sum of squares within. And then we, in the single factor ANOVA, we would have had the treatment. But in this case now, we have two treatments. So we have, in this case, we have sums of squares, the variation due to fat type. We have the sums of squares due to sex, and we have the sums of squares due to the fat type by sex interaction. This is all what in a one-way ANOVA would have been sum squared treatment. So this is essentially the way an analysis of variance gets structured. We have a total amount of variation. How much of that variation is due to systematic variation due to our treatments? and how much of that is just random noise. Now, there's a certain amount that is random noise, and then this systematic treatment, is that systematic variation due to fat type? Is it due to sex? Or is it due to some interaction between the two? So all of these things should add up to the total amount of variation. This is pretty easy to calculate because we already know how to calculate that. This is pretty easy to calculate because we already know how to calculate that. We've done these two things for a single factor ANOVA. The question in a two-way ANOVA is, how do we quantify these other things? And that becomes a little more complicated. So let's start with what we know, which we know how to calculate sum squares total, and we know how to calculate sum squares within. If we took 709 minus that, squared that difference, 679 minus that, squared that difference, took this difference and squared it, added those up, that would be 466.67. If we took 592 minus 535 and squared that difference, squared this difference, added those up, that would be 6,738. Did the same thing here, here, and here, that would be 3752. And then did here, here, and here, that would be 708. Add all of that up, that's 11666.67. That is the sum squares within. That's no different than what you would do in a single factor analysis of variance. It's just the within sample variation. If we subtract this from that, that tells us then if we take 7757025 and subtract from that 11666.67, that would give us 65903.58. So let's do that. That would be 8. We would borrow that from there, would that be 9? 5, 3, that 9 gets borrowed from that 6 though, right? 0, am I doing my math right? Yep, I am doing my math right. 15 comes from there, so this would be 6, 6, 5, 9, 0, 3, 5, 8. 6, 5, 9, 0, 3, 5, 8. So if we take the total sums of squares, subtract from that the within sample variation, it gives you the sums of squares of subgroups. That's the total amount of variation. This whole thing here should all add up to 65903.58. Because if it's not random variation within samples, it's a variation that is due to your treatments. So this whole thing should ultimately add up to this. This is one way of calculating this, but just like everything else in statistics, there's more than one way to do this. We could also take each of these means, this one, this one, this one, and this one, subtract those from the grand mean, add up all those squared deviations, and it should give us the same thing. So your homework for Wednesday is do that calculation, see if it gives you the same answer that you get when you subtract total sums of squares from within sum of squares from the total sum of squares, and it should give you the same thing. Because we're out of time, and we'll pick up here next time.